Good morning, everyone. I hope everyone has been enjoying the meeting. I know that I have. My name is Abena Brewster, and I'm a professor in the Department of Clinical Cancer Prevention at MD Anderson Cancer Center. And today I have the great privilege to introduce the 15th recipient of the AACR Outstanding Investigator Award for Breast Cancer Research. Since 2008, the AACR has been distributing this special award to recognize investigators no more than age 50 whose novel and significant work has had or may have a far-reaching impact on the etiology, detection, diagnosis, or prevention of breast cancer. Such work has involved numerous disciplines across the continuum of biomedical research, including basic, translational, clinical, and epidemiological studies. Since 2018, the Breast Cancer Research Foundation has generously supported this special award, which continues to provide an important platform by which to recognize and honor emerging leaders in the field of breast cancer research. Please join me in thanking the Breast Cancer Research Foundation for their ongoing support. So this year's award recipient, Dr. Adentunji Toriola, continues the rich tradition of this award to recognize impactful scientific achievements in breast cancer research. Dr. Toriola is a tenured professor of surgery in the Division of Public Health Sciences and the William H. Danforth Physician Scholar at the Washington University School of Medicine in St. Louis, Missouri. He also co-leads the Cancer Prevention Control Program at the Siteman Cancer Center. He is being recognized for his effort in combining his expertise in public health, epidemiology, and medical training to identify and evaluate strategies for breast cancer research. Through paradigm-shifting innovative research, Dr. Toriola demonstrated the relationship between the rank pathway and mammographic breast density in premenopausal women. Subsequent studies demonstrated that circulating rank biomarkers are also associated with mammographic breast density. Having provided the first evidence that rank signaling is a viable pathway to target in reducing mammographic breast density and the development of breast cancer in premenopausal women, Dr. Toriola developed a phase 1 clinical trial that demonstrated that a single 60 milligram dose of denosumab is capable of altering pathways involved in hormone, immune, and inflammatory signaling in the breast tissue of healthy premenopausal women with dense breast, setting up the potential utility of denosumab as a chemoprevention agent in high-risk women. Notably, Dr. Toriola has successfully established a platform by which to rapidly translate preclinical and clinical research findings into preventive therapy clinical trials, and is currently translating this phase 1 clinical trial finding into real-world application through innovative phase 2 clinical trials. This research has fulfilled an unmet need in the prevention of breast cancer in premenopausal women, given the limited number of prevention options available to premenopausal women, who account for approximately a quarter of all new breast cancer cases diagnosed annually in the United States, and even larger globally. Collectively, Dr. Toriola's research holds great promise for the future of breast cancer research, and I am thrilled to introduce him as this year's recipient of the AACR Outstanding Investigator Award for Breast Cancer Research.
Thank you, Dr. Brewster, for that very kind introduction. I would like to thank AACR and Breast Cancer Research Foundation for this privilege and honor recognizing our work. I'll be taking us through some of the work we've done over the past 10 years in terms of preventing breast cancer and identifying new biomarkers of premenopausal breast cancer prevention. Worldwide in 2020, more than almost a quarter of the new breast cancer cases diagnosed were in premenopausal women. In the United States, the incidence of breast cancer is increasing, especially in premenopausal women. If you compare the cohort effect, there's been an increase in the birth cohort effect, with women born in the younger cohorts of more recent years having a 20% increased risk of breast cancer compared to women born in 1955 cohorts. This indicates that we need to double up our efforts at primary prevention of breast cancer, especially in premenopausal women. My talk is going to focus on some of the things that we have done over the past 10 years. While population-based prevention methods are very valuable and very effective, I'll focus on some of the paths that we have taken, and these are the opportunities that I see for prevention, primary prevention of premenopausal breast cancer. We need to identify novel biomarkers and pathways that can be targeted in premenopausal breast cancer prevention. We need to expand chemoprevention options for high-risk women. We need to give greater focus on high-risk women who do not have mutation in high-risk breast cancer genes, and there's an opportunity to target intermediate phenotypes. And what better intermediate phenotype to target than mammographic density? As you can see, compared to all the other breast cancer risk factors that confer a greater than 4-fold increased risk of breast cancer, mammographic density is potentially one that is modifiable. When it comes to populatory attributable risk factors for breast cancer, mammographic density confers one of the largest attributable risks for breast cancer. 29% of breast cancer cases in premenopausal women is thought to be due to women having extremely dense breast or heterogeneously dense breast. This implies that in terms of primary prevention, if we reduce women's dense breast from highly dense to non-dense breast, we could potentially reduce 28 to 29% of breast cancer cases in premenopausal women. In addition to being a strong risk factor for breast cancer, high mammographic density also share similar biological and genetic pathways with breast cancer risk, further strengthening the need to target mammographic breast density in breast cancer prevention, especially in premenopausal women. And observational studies have shown that a decrease in breast cancer mammographic density over time translates to a decrease in breast cancer risk over time. This is a recent meta-analysis that was published early this year and shows that compared to women whose density stays the same over a long period of time, women who have a decrease in breast density, non-pharmacological decrease, have a 22% reduction in breast cancer risk. However, the biological basis of mammographic breast density are poorly understood, and we have limited knowledge on how to reduce dense breast to decrease breast cancer risk. We have been doing a lot of work to understand drivers of mammographic breast density in premenopausal women, and we've observed that about 25% of the variance in mammographic breast density can be explained by adiposity. Alcohol consumption is weakly associated with mammographic density. There is no convincing evidence linking physical activity to mammographic density yet. Hormone replacement therapy use is a driver of mammographic density in postmenopausal women, and there's been no convincing evidence linking adult diet to increase mammographic density so far. Family history of breast cancer is also a strong driver of high mammographic density. So while population-based preventive measures have been known to be very effective in preventing and reducing breast cancer risk, adopting sustained healthier lifestyle for breast cancer prevention is very challenging. And this is a review done by Graham Colditz and Kathy Bucket that was published about 8 years ago. And you can see why the time until benefit for most of the lifestyle factors to take effect in preventing breast cancer tends to take a long time, between 10 to 30 years. But on the other hand, when it comes to targeted prevention in specific high-risk individuals, the time to benefit tends to be shorter. For instance, the effects of tamoxifen can be glimpsed as early as 2 years from initiation of tamoxifen. So this prevents us an opportunity to use targeted prevention in high-risk specific populations. We recently published a systematic review looking at chemoprevention trials and their impact on mammographic breast density in premenopausal women who do not have breast cancer. We identified 7 clinical trials looking at 6 different agents, and what we found was that only one agent, which is tamoxifen, has been shown to have a clinically significant decrease in mammographic breast density. The gonadotropin receptor analogs also reduce mammographic density over time, but these were not clinically significant. And this is the data from the IBIS trial showing the impact of tamoxifen on mammographic density and breast cancer development. In this clinical trial run out of England, it was noticed that tamoxifen reduced mammographic density as early as 12 months. This reduction in mammographic density translated to a reduction in breast cancer risk at the 6-year time point. What was most striking about this study was that only women who were assigned to tamoxifen, who had a greater than 10% reduction in mammographic breast density, benefited from the chemopreventive effect of tamoxifen. Women who were assigned to tamoxifen but had a less than 10% reduction in mammographic breast density did not benefit from the chemoprevention effect of tamoxifen. However, tamoxifen targets only estrogen-related pathways. Less than 0.3% of women in the US who are eligible to use tamoxifen for chemoprevention use it because of its side effects. We therefore need to identify new pathways that have fewer side effects, that have broader applicability, especially in premenopausal women. A potential target is estrogen plus progesterone. Studies within the Women's Health Initiative have shown that women assigned to estrogen, women who use estrogen plus progesterone have an increase in mammographic density within 2 years of starting the estrogen plus progesterone use. It was also noticed that these women went up, estrogen plus progesterone use was related to an increased risk of breast cancer. What was one of the strongest findings from this study was that a 1-year increase in mammographic breast density predicted subsequent development of breast cancer, and all the breast cancer development was mediated through an increase in mammographic breast density. So this brings us to the question of potentially targeting progesterone signaling or downstream mediators for progesterone signaling in mammographic breast density and breast cancer risk. Several labs have identified an important role for rank ligand signaling in progesterone signaling. Rank ligand signaling mediates the major proliferative response of progesterone to mammary epithelial cell and drives the major action of progesterone on mammary epithelial cell cancer development. In addition to being a strong driver of mediator of progesterone signaling, rank ligand signaling also mediates other pathways that are involved in breast cancer development. Over the last few years, we have been looking at the roles of rank ligand signaling in mammographic density through a series of observational studies, and also we've completed a phase 1 clinical trial, and we're currently enrolling for a phase 2 clinical trial now. I'll go through some of our findings over the past few years. In the first study, which was a pilot study in 48 women who had provided breast tissues to the St. Louis Breast Tissue Registry, we used NanoString Encounter platform to profile breast tissue gene expression. And what was very striking was that even though the study was a small study in 48 women, rank ligand signaling and tumor necrosis factor gene expression were positively associated with mammographic breast density in these women. We used BIRAT classification for our mammographic density. This was exciting, and it gave us the encouragement to go ahead to do a prospective observational study. For this study, we recruited 365 premenopausal women who came in for annual screening mammogram at Siteman Cancer Center. On the day of their mammogram, we collected blood sample and questionnaire-related information on breast cancer risk factors. When we analyzed their blood samples, we found out that women who had higher circulating rank ligand and rank ligand concentrations had higher volumetric percent density and also higher dense volume. In this overall analysis within the whole study population, we did not find an association between circulating soluble rank ligand and volumetric percent density and dense volume. But based on our understanding of the biology of relationship between rank ligand pathway and progesterone signaling, we decided to stratify our analysis by circulating progesterone levels. We discovered that among women who had higher progesterone levels, both circulating rank concentration and soluble rank ligand concentrations were positively associated with volumetric percent density, but these findings were not replicated among women with low circulating progesterone level, setting up the stage for looking at rank ligand signaling and among women with high progesterone levels. Based on this encouraging findings, there is a finding in premenopausal women which is just published, but I've not included that here. So basically, we confirmed some of the findings that we found in premenopausal women in 400 postmenopausal women. We also found very strong associations and correlations between plasma rank ligand gene expression and progesterone and prolactin as well, which is potentially an interesting target for future studies. So clinical trials on rank ligand inhibition and breast tissue markers, we've just completed a phase 1 clinical trial, and like I mentioned, a phase 2 clinical trial is currently ongoing. For the phase 1 clinical trial, we identified 10 premenopausal women who had dense breast, and more than half of them, 6 of them, had a positive family history of breast cancer. At baseline, we took breast biopsy and blood draw. We then gave them 1 subcutaneous dose of 60 milligram denosumab. This woman then came back at 60 days, and we tried as much as possible to time the return biopsy and blood draw within the same phase of the menstrual cycle. So they had a repeat breast biopsy and blood draw at 60 days. We observed that rank ligand inhibition or denosumab downregulated pathways involved on immune regulation, inflammatory signaling, particularly macrophage markers including CCR5, CD85, and CD300A, and targets downstream of interferon 8 were downregulated in these women. We observed that pathways involved in progesterone metabolism, estrogen response, and fatty acid metabolism were upregulated on day 60. In particular, we found very strong downregulation of UDP glucuronide transferase family 2 member 15, which plays critical role in regulation of estrogen and androgens, and also in alpha aldoketoreductase family 1 member 15, which enables estradiol 17 beta dehydrogenase activity. We then further evaluated our analysis using the comprehensive multi-omics platform for biologic interpretation developed at the McDonald Genome Institute at Washington University School of Medicine. And our findings were identical. We found upregulation and downregulation in signaling pathways related to hormone signaling, estrogen response, inflammatory and immune signaling. We also tried to see whether there is an overlap between the biology of rank ligand inhibition within the breast and within the blood plasma as well. Working with Chris Mayer and his group, we found limited overlap in terms of the effect of rank ligand inhibition on breast tissue gene expression and blood gene expression. Based on this exciting preliminary data, we were funded by the NCI to perform a phase 2 clinical trial looking at the impact of denosumab on mammographic density in premenopausal women. And to be eligible for this study, we recruited premenopausal women more than 40 years who had dense breast on mammogram, and they could be at an increased risk of breast cancer. Our goal is to recruit 210 women, randomize half of them to an intervention arm of denosumab, and the other half to an intervention arm of placebo. The goal is to give them 2 doses of subcutaneous denosumab, 60 milligram at baseline, and also at 6 months, and also the placebo arm as well, 60 milligrams, 2 subcutaneous doses of placebo at baseline and at 6 months. Our primary outcome measure is change in mammographic density at 12 months, and our goal is to compare change in the denosumab arm to the placebo arm. We are also measuring mammographic density in addition to baseline measures, also at 12 months and at 24 months. We are performing breast biopsy on all study participants at baseline at 12 months, and also collecting blood samples at baseline and at 12 months. Our secondary goals are to look at breast tissue gene expression and compare the impact of rank ligand inhibition, also look at blood gene expression as well. Depending on if our findings are positive, we hope to build a predictive marker to determine who will respond to denosumab intervention. These clinical trials, phase 2 clinical trials started in September 2019. So far, we've been able to recruit 117 premenopausal women onto the study. And even though we've consented more women, we've consented about 155 women so far, but only 117 of them are currently on the trial. 5 of those women are currently in screening. We discovered that a few women were ineligible after we had consented them. We discovered that they were ineligible after we performed hormonal testing. Even though from their medical history, they appear to be premenopausal, but after doing hormonal testing, we discovered that they were postmenopausal, so we excluded them from the trial based on that. And so far, only 3 women have been lost to follow-up, and 4 women have withdrawn from the study. 12 women are currently enrolled to be re-enrolled and reconsented because they were enrolled during the height of COVID pandemic, and they promised to get back during their next phase of their next mammogram. We are very passionate about ensuring underrepresented minorities are enrolled in clinical trials at Sightman Cancer Center. So far, 76% of study participants in this clinical trial are non-Hispanic white women, and 25% are underrepresented minorities, of which 19% are non-Hispanic black women. As of November 30, 2022, we have completed baseline mammogram on 119 women. We have completed blood draw and baseline intervention and biopsy on 117 women. A total of 101 women have gotten to the 6-month intervention time point, and we've already completed the 6-month intervention visit on 97 women. 72 women have completed 12-month mammogram, and 79 women have completed baseline mammogram, and 72 women have completed 12-month blood draw and biopsy. This is the risk profile distribution of women enrolled in the trial. 48% of them have an above average risk of breast cancer. 30% have a much above average risk of breast cancer, and 22% have an average risk of breast cancer. We have been using the Rosner-Colditz model to determine women's risk of breast cancer, and this is incorporated into some of our clinical activities at the Breast Health Center at Sightman Cancer Center. We have also been taking adequate care to follow up and look at adverse events in these women. So far, we have not experienced or recorded any grade 4 or grade 5 adverse events. Most of the adverse events that we've reported are grades 1 and 2, and they've been related to muscle cramps and periodontal pain, but these are very transient and they resolve quickly over time. Because this is a very large undertaking and endeavor, my team has screened over 11,000 women so far since this trial began in 2019. Of these 11,270 women, about 24% are eligible for the clinical trial. And for those who are eligible that chose not to participate, we've been trying to find out reasons why so that we can better incorporate some of these reasons into future clinical trials. And for majority of them, about 72%, they just don't want to be involved in a clinical study. 10% cited not wanting to take denosumab as a reason for not participating, and about 6% cited the need for biopsy as a reason for not being a part of the trial. In summary, close to 25% of breast cancer cases are diagnosed in premenopausal women, but lifestyle-related factors to reduce risk and chemoprevention options in premenopausal women are limited compared to postmenopausal women. Currently, only one chemoprevention agent has been approved for chemoprevention in premenopausal women compared to 6 in postmenopausal women. Almost 29% of breast cancer cases in premenopausal women can be attributable to having dense breasts. This is data from the Breast Cancer Surveillance Consortium. Yet we have limited knowledge on how to reduce breast cancer development in high-risk women with dense breasts. Studies from our team has convincingly shown that both breast tissue rank ligand gene expression and circulating rank biomarkers are positively associated with mammographic density in premenopausal women. We've completed a phase 1 clinical trial that demonstrated that rank ligand inhibition resulted in regulation of signaling pathways essential for hormone metabolism, proliferation, inflammation, and immune regulation. Our phase 2 clinical trial is ongoing, and successful demonstration that denosumab reduces mammographic density in premenopausal women could open up additional approaches to primary prevention in select high-risk premenopausal women with dense breasts. In addition to our work on rank ligand signaling and mammographic density, we've also begun to apply other genomic approaches to interrogate mammographic breast density and take that to breast cancer prevention as well. Recently, we recruited 705 premenopausal women into an observational study, collected blood samples from them and on the day of their screening mammogram, and also collected detailed data on breast cancer risk factors. We completed lipidomic profiling of 982 lipid species in 14 lipid pathways. And these are still very preliminary findings. Detailed analysis are still ongoing. But we're beginning to discover that lipid pathways in pathways exoceramide, lactoceramide, and lipophosphatidylcholine may be positively associated with mammographic density. This is not surprising given that 25% of the variance in volumetric percent density is mediated by adiposity. After very stringent Bonferroni correction, we discovered that one particular lipid species, which is phosphatidylinositol 18-1-18-1, is positively associated with volumetric percent density. This is a lipid species, an adipocyte lipid species that limits stress signaling in preclinical models. We've also been applying metabolomic profiling on these 705 women, and we've been able to identify a metabolite profiles or metabolite species that differentiate women with dense breasts from those with non-dense breasts with almost 95% or 97% accuracy. I would like to highlight a few of these metabolites. These are progesterone metabolites and also other androgenic metabolites. As you can see, there are very strong positive associations between some of the progestin metabolites and also pregnenolone metabolites and mammographic density. But one of the intriguing findings is that androgenic steroids, depending on what they are, could be positively associated with mammographic breast density as well as inversely associated with mammographic breast density. Some of our initial findings also relate to gut-associated metabolites being positively associated with mammographic breast density, as well as markers of collagen formation and tissue remodeling as well. One particularly notable metabolite in tissue remodeling that we've observed, which has a very strong association with volumetric percent density, is hydroxyproline. Our goal after completing all this analysis, looking at these lipid profiles and metabolomic profile and mammographic density, is to take our findings to breast cancer and determine whether these lipid profiles and also these metabolomic profiles that predict having dense breasts can also predict having developing breast cancer risk in premenopausal women as well. In summary, I'd like to conclude with the following. There is an urgent need to mitigate the rise in the incidence of premenopausal breast cancer. This will require us to continue to adopt the population-based approaches, which we have been doing all along, but with understanding the potential limitations and long-term time to benefit. And we can also adopt precision prevention approaches as well. And we started doing that by applying genomic methods to identify additional pathways that can be targeted for prevention in premenopausal women. There is a need for the field to develop novel agents that can be personalized according to risk pathways, especially in women who do not have mutation in high-risk breast cancer genes. We also need to start looking at rational drug repurposing for new chemoprevention agents that can be applied across board, that have limited side effects, that are long-lasting, and that are not related to cancer risk in the public mind. We need to develop platforms to rapidly translate epi and clinical findings to chemoprevention clinical trials. And a lot of this work would not have been possible without really detailed and focused transdisciplinary collaborations, which we need to keep encouraging. Finally, I would like to thank these collaborators and co-investigators who have made this work possible. I'd like to pick out Graham Coldis, who has been a great collaborator and mentor over the past 10 years. I'd also like to thank our patient advocate, Judy Johnson, who has provided patient perspectives on all these studies that we're doing. Also, I have a very wonderful program officer who has been very supportive and who has provided excellent feedback along the way. I'd like to thank members of my team who have made this work possible, and our funding sources from Susan G. Komen Foundation, the NIH. I'd like to highlight that Susan G. Komen Foundation made the initial grant that allowed us to go in this pathway possible. Thank you very much for your attention.